Hello, everyone, and thank you for choosing to attend our virtual conference session today. I'm personally very excited about the advancements being made in the areas of immuno-oncology and cell therapies, so I've been eagerly anticipating this presentation by our speaker. Dr. Hongbo Chi received his Ph.D. from the University of Rochester and subsequently received his postdoctoral training from Yale University School of Medicine. In 2007, he started his independent research program at St. Jude's Children's Research Hospital, where he's currently a member and professor in the Department of Immunology and the Robert G. Webster Endowed Chair in Immunology. His research focuses on mechanisms of immune signaling and metabolism, in particular how mTOR and other signaling pathways interplay with metabolic programs of T-cell fate decisions. His laboratory has discovered the critical importance of mTOR signaling, metabolism and ontophagy in the differentiation and function of effector and regulatory T-cells, the control of T-cell quiescence and antigen-triggered exit from quiescence, as well as the regulation of autoimmune and infectious diseases and cancer. His research also has contributed to our understanding of the signaling and metabolic pathways in dendritic cell biology and the crosstalk between innate and adaptive immunity. More recently, Dr. Chi's laboratory applies systems biology and immunology approaches such as proteomics, metabolomics, single-cell RNA sequencing, and CRISPR screening and integrative network analysis to reconstruct metabolic signaling circuits and identify new therapeutic targets in cancer and other immune-mediated diseases. And now, having put all of that out there for you, I'd like to hand it over to Dr. Chi to walk us through some of his presentation of some of his findings.
Hello, everyone. It's great to be here today, and I want to thank Agilent for the opportunity to speak today. The research program of my lab is in immunometabolism, especially the interplay between signaling pathways and the metabolic programs for the control of immunity and tolerance. We are particularly interested in several metabolically related kinases, including mTOR, LKP1, and Hippo signaling. On the metabolic side, we are interested in understanding cell bioenergetic activities like glycolysis and oxidative phosphorylation, the cell biosynthetic activity to build biomass, and as well as nutrient sensing, how nutrient can impact immune signaling. So therefore, we'll consider this as a bidirectional metabolic signaling. Within this underlying theme, we explore T-cell biology as the main focus, and we also investigate how T-cell responses are controlled by the antigen-presenting cell, dendritic cell, and dendritic cell metabolism. For today's discussion, I'll be mainly focusing on work in T-cell immunometabolism. First, I'd like to give an overview of our research, especially our more recent approaches that rely upon the integration with system immunology. Then I'll be sharing with you two stories, one on Treg cells and the other one on CD cells, and also the role of this immunometabolic pathways in immune responses and anti-tumor immunity. Metabolism, as we understand, is a fundamental requirement of all living cells. But instead of simply serving as housekeeping function, metabolic activity are actively regulated. For example, for proliferating cells or tumor cells, they will highly engage glycolysis even in the presence of abundant oxygen. Therefore, this phenomenon is also known as aerobic glycolysis or Warburg effect. For activated T cell, partly because their massive rate of proliferation, it's not surprised that they will upregulate glycolysis as compared to naive T cell. However, a number of years ago, in collaboration with Doug Green Group, we had this somewhat surprising finding that not only is glycolysis upregulated upon T cell activation, it is differentially regulated when naive T cells develop into effector cells, Th1, Th2, or Th17 cells, compared when they differentiate it into induced Treg cells. And we further show that the Th17 differentiation is partly dependent upon glycolysis and the transcription factor P1 alpha. So highlighting the important role of metabolism in cell fate control. Shown as this nice review from Edgar Pierce, metabolic pathway has been identified in multiple stages and multiple cell types involved in T cell biology. As a matter of fact, metabolism is essentially involved in every aspect of T cell biology, starting from thymocyte development to peripheral T cell activation, and then for the activated cell to transition into this effector subset or regulatory subset, or for they to differentiate into memory cells. In this recent review in Nature Review Immunology, we described several of these intracellular metabolic programs, including the glycolysis, mitochondrial metabolism, and lipid synthesis, for instance. We also highlight the importance of the control of metabolism by the surface receptors and these nutrient transporters. This is also dependent upon the interaction and crosstalk between T cells and other cells, such as T cells. Among the central regulators of immunometabolism is mTOR. mTOR is a kinase. It's got the name from the initial discovery of the immunosuppressant rapamycin. And therefore, mTOR stands for mechanistic target of rapamycin. mTOR functions through one of the kinase complexes, mTOR1, that can be defined by the molecule Raptor, or mTOR2, and the Richter molecule as the adapter. And for the upstream, many of the upstream molecules are important to control mTOR activity either positively or negatively. A unique feature of mTOR, especially mTOR1 activation, is its ability to integrate multiple upstream signals, including growth factors, immune signals, and nutrients. Over the past few years, my lab has been actively investigating the role of mTOR in T cell biology. On the one hand, we're particularly interested in how mTOR controls T cell quiescence, its activation, and fate decisions. On the other hand, we're also interested in how mTOR signaling contributes to Treg cell biology. Well, we like to draw the metabolic program as linear arrows and simple cartoons, but in reality, metabolism functions as highly complex metabolic network with extensive crosstalk, feedback, feed-forward loops, and metabolic adaptation. So therefore, we feel studying one molecule at a time is no longer sufficient, and system-level knowledge is essential to integrate the information for better biological insight. System knowledge refers to use the large data set and unbiased approaches to integrate the knowledge, the biological information. And so in this case, of course, this rely upon the profiling of metabolomics, transcriptomics, and other omics at both the population level or single cell level. But more importantly, it's the integration of such information that is critical to gain biological insight. So as a matter of fact, there's increasing appreciation of the power of combining the system approaches and the reductionist or one molecule at a time approaches to dissect complex biological traits. So therefore, in the next few minutes, let me just show one example of how my lab is combining system immunology and reductionist approach to understand mechanism of the metabolic activation in T cells. T cells, as we know, can be largely divided into the quiescent or non-quiescent cells. For the quiescent T cell, they are very small. They are in the T0 cell phase of cell cycle, and also they use mainly the oxidative metabolism from their antigen energy source. But once T cell receive anti-stimulation, they grow, they upregulate glycolysis, as I mentioned, and also they have a unique anabolic metabolism. What's driving this transition from naive to activated T cells has remained unclear, but it's a long-standing interest in my lab. As shown by this recent cover of Nature Review Immunology, is interesting for us to understand how we can wake up T cells, how we can wake up naive T cells through tuning of metabolic processes. Our journey in this area started when we made a T cell specific knockout of TSC1, the tumor suppressor. And we found that T cell deficient in TSC1 have increased cell size, activation markers, and cell cycling, suggesting that they have lost their maintenance of cell quiescence. As a consequence, the cell tend to undergo apoptosis. Mechanistically, this is due to the aberrant activation of mTOR1, so suggesting that mTOR1 need to be actively suppressed by TSC1 to enforce naive T cell quiescence and immune homeostasis. Based on this observation, we next ask, what's the functional importance of mTOR1 activation upon antigen stimulation? Therefore, we generated mice deficient in Raptor or mTOR1, Richter or mTOR2 or both, and challenged those T cell specific knockout mice with Listeria infection. We found that Raptor deficiency essentially abrogated antigen specific T cell responses upon Listeria infection. In contrast, Richter or mTOR2 deficient showed only a modest effect in response to Listeria infection. So therefore, highlighting the critical importance of mTOR1 activation in driving T cell priming and antigen stimulation-induced exit from quiescence. Based on this observation, we were interested in understanding the additional pathway and signaling circuits that are important to drive naive T cell exit from quiescence. So therefore, we teamed up with Dr. Jim at St. Jude and did a proteomics profiling of naive T cells or T cells stimulated at different time points. We did the whole protein and phosphoprotein profiling, followed by network analysis of this multi-OMICS data. This led us to identify several mitochondrial pathways that are important to drive T cell exit from quiescence. So therefore, although glycolysis is upregulated upon antigen stimulation, remodeling of mitochondrial pathways is critical to mediate the exit of T cells from quiescence. Based on both this genetic and proteomic study, we proposed the following model. We proposed the 6 hallmarks of naive T cell exit from quiescence, including the cell cycle entry, cell growth, the cytokine signaling, anabolic metabolism, nutrient uptake, and the reprogramming of mitochondrial function, with mTOR1 as a central player to coordinate extracellular events and intracellular metabolic reprogramming. Moreover, we can apply this principle of the immune quiescence and activation to other contexts, including the T17 responses. And we can show that mTOR is important to direct the two fates of T17 cell, either the quiescent, the stem-like cell fate, or the terminal differentiated Th1-like T17 cells. Moreover, we are interested in understanding how T cell priming and T cell exit from quiescence can be directed by the signals from dendritic cells. And we show that, for example, the key role of people signaling in mediating the CDC1 activity to drive T cell priming. So with this brief summary of our work in system immunology, let me start to focus on our next story, the Tregs and the nutrient sensing and signaling. Regulatory T cells, as we know, are powerful cell types for immune suppression. Regulatory T cells or Treg cells are important to inhibit autoimmune responses under steady state, the lack of which would result in the development of autoimmune diseases. But on the other hand, Treg cells are a major barrier to effective tumor immunity and tumor immunotherapy. Not all Treg cells are created equal, in that Treg cells have discrete activation profiles. The resting of the central Treg cells can also sometimes known as the R-Treg resting of central Treg. Upon antigen stimulation, they can develop into the effector Treg cells, also known as A-Treg or E-Treg cells, and this cell can further populate the non-lymph organs known as tissue-specific A-Treg cells. Our work in this area is partly because of our initial funding. That's surprising at that time because although mTOR has been generally considered as a natural regulator of Treg cells, we were surprised to find that when we knock out mTOR1 or raptor molecule from Treg cells, mice die from inflammation. This highlights a critical positive role of mTOR1 in programming Treg function. And mechanistically, we show that mTOR is important to coordinate the Treg cell proliferation and their movalent activation of the movalent pathway in driving the expression of Treg cell effector molecules, and altogether contributing to programming the Treg suppressive function. This data highlight the critical role of mTOR as a positive regulator. But then we ask, is more better? To address this question, we knock out Pten, which is critical negative regulator of mTOR. And we found that mice with Treg-specific loss of Pten develop a lupus-like symptom. And this is because Pten is important to keep mTOR, especially mTOR2, in control. And when the Treg cell lose Pten, they become destabilized, and the FAP3 expression is also dampened. So therefore, Pten is critical to keep Treg function intact. Based on this data, we show that either too little mTOR1, either too little mTOR activity, as shown by the raptor or mTOR knockout, or too much mTOR activity, as shown by Pten or ATG7 knockout. In either case, is detrimental to Treg-mediated immune tolerance. Therefore, we come up with this model showing it's the Goldilocks principle to explain mTOR signaling in Treg cells, as shown here, mTOR1 or mTOR2, and either too much or too little of these activities is effective for in regulating Treg cell function and their lineage stability. having this finding, we are also interested in understanding how this mTOR activity regulated. As shown by this diagram from Michael Hall a couple of years ago, this upstream regulation is a highly complex process. This elegant work from Michael Hall, David Sabatini, and others that have mapped multiple molecules, some of them as nutrient sensors, to activate mTOR1. This upstream signals is very little information in T cells or Treg cells. And so for us, many of them actually identified in the cell line may not necessarily function in T cells. For example, we show the LPP1 function is important for Treg cells, but it's through a AMPK or mTOR independent process. Therefore, we are quite interested in understanding all these upstream signals involved in activating or inhibiting mTOR activity. Here we can just focus on this two groups of small T protein, the RAG small T protein, or the RAG small T proteins, as well the nutrient sensors, because arginine, leucine, and glutamine has been shown to be important for mTOR activation. This project, Hongbo and Nicole, very talented fellow in the lab, took the challenge. We first studied the role of amino acid, which are the classical signals to activate mTOR. Here we compare the ability of naive T cells and Treg cells for their responsiveness to amino acid stimulated mTOR activity, as shown here by the phosphorylation of S6. As you can see, naive T cells have a low baseline mTOR activity, which does not get much upregulated upon amino acid stimulation. In contrast, Treg cells have a higher, a little bit higher baseline mTOR activity, but this can be further upregulated upon amino acid stimulation. We further show that this increased mTOR activity is associated by the higher expression of certain amino acid transporters on Treg cells compared with the naive T cells. This includes CD98, which can transport leucine, and also the SLC7A1, which can transport arginine. Moreover, we stimulated resting Treg cells with CD3, CD28, and plus L2 overnight. This basically will drive Treg cells to be activated. We call them TCR-activated Treg cells or A-Treg cells for simplicity. We found that these A-Treg cells compared with resting Treg cells have higher expression of these nutrient transporters, these amino acid transporters. Also, the A-Treg cell have much higher intracellular levels of multiple amino acids. And also, not a surprise, this TCR stimulation also results in a strong induction of mTOR1 in A-Treg cells. Given all this information, we next design an experiment to screen for which type of amino acid are important for A-Treg cells. To this end, we use the Biolock, this nutrient screening system. Each of the well contains a single amino acid or a combination of amino acid. And then we use this overnight activated Treg cell as our cell type for screening. And you can see here, we found that, to a surprise, the nutrients that are able to induce higher mTOR activity, they all contain arginine. This arginine with another amino acid. So highlighting probably arginine has an important role in mTOR1 activation. Built upon this information, then we did the following validation experiment. First, we stimulated this A-Treg cell with different amino acid or some of the different amino acid combination, and we found that arginine has strong effect to upregulate mTOR1 activity. And also arginine plus leucine also has further ability to upregulate mTOR1. On the other hand, the glutamine, although has been shown to be able to upregulate mTOR in other cells, we do not find a strong role in this system of the activated Treg cells. One of the mechanisms for mTOR to be activated is its translocation to lysosome. So therefore, we measured whether amino acid has a role in this process in Treg cells. As you can see, without amino acid stimulation, the mTOR, the red color, or lysosome, the green color, are largely discrete. But with amino acid stimulation, the proportion of them co-localized, as can be shown by the co-localized yellow signal. Well, stimulation with interleukin-2 cannot do so. So therefore highlighting a specific role of amino acid in driving mTOR translocation to lysosome. Having shown the importance of amino acid, especially arginine, in this process, we next studied used to do the reverse experiment. We have the Treg cells are activated with full amino acid, then we deprive a single amino acid. We found that deprivation of arginine alone was able to substantially downregulate mTOR activity. And also deprivation of leucine can also have an effect, although the downregulation effect is less profound compared with arginine. On the other hand, again, glutamine deprivation had no strong effect. And this importance of arginine or leucine in mTOR1 activation can be also appreciated in vivo, in that the mice have the leucine diet or arginine diet and are found to have reduced mTOR-dependent expression of this CTR4 and ICOS on Treg cells. So everything together, we conclude that arginine and leucine are the major regulators of mTOR activity in Treg cells. Having shown this information, we next study the interplay between this amino acid signal and the TCR signal. Because after all, TCR signal are known as the signal one, the most important signal to drive T-cell activation, and also including mTOR1 activation. So here we did this experiment. We stimulated Treg cells with TCR. Then in the absence or presence of amino acid, we found that the TCR is able to activate mTOR1 only in the presence of amino acid. Well, when the T cell receive TCR signal without amino acid or with amino acid washed off, their ability to activate mTOR1 is essentially abrogated. So that's suggested the importance of amino acid to enable TCR-dependent mTOR1 activation. So we also call this a role of licensing role. And this licensing role by amino acid can be also evidenced by the examination of the TCR-dependent activation events. Basically, after overnight stimulation with TCR, there's upregulation of CD7, the surface molecule, as part of the activation events, and also there's extensive increase in cell size. But we found that when Treg cells are stimulated with TCR without amino acid, these activation events were greatly reduced. So therefore, we conclude that amino acid has this licensing role to support TCR-dependent mTOR activation. So, and also going back to this cartoon, we show especially in this process, arginine and leucine are critical, but glutamine does not seem to be so. Having this information, we next dissected the downstream molecule that can link the amino acid to the activation of mTOR1. And here we first focus on the RAG-A, RAG-B, the multi-proteins, and we obtained the flux mice from Kunliang Guan group at UCSD and made the CD4 conditional knockout mice. As you can see here, the white caps Treg cell from this mice can respond to either the arginine leucine or the full cocktail of amino acid by upregulating mTOR1, but this ability was greatly lost in REG-AB knockout Treg cells. And also using seahorse bioanalyzer, we found that upon TCR stimulation, the oxygen consumption rate and the glycolytic rate are also both greatly reduced in Treg cells lacking REG-A, REG-B. So therefore, the REG-A and REG-B are important for mTOR1 activation driven by TCR and amino acid stimulation. To further understand the critical role of this molecule in Treg cell biology in vivo, we generated a separate knockout mice by crossing this FOX allele with FOXP3, allowing us to deplete this molecule in Treg cells specifically. And we found that in this case, the mice with Treg specific knockout, REG-A and REG-B, they have the systematic inflammation as shown by the splenectomy, and also the increased lymph node, and also ultimately the mice will die from the inflammation. As you can see, there's extensive infiltration of leukocytes and immune cells in multiple organs, including colon, liver, and the lung. And also between these two molecules, these are the homolog molecules, these two small G proteins, we found that the REG-A deficient mice also develop a fetal disease, but at a later onset. Or the REG-B knockout mice have no substantial phenotype. So therefore, this gives us the opportunity to study the role of this pathway in Treg-mediated anti-tumor immunity. To this end, we challenged either the wild type or the REG-A single knockout mice with the MT38 transformer tumor model. And we can see the REG-A Treg knockout mice can show much better ability to control tumor growth. This is also consistent with the single-cell RNA-seq analysis of the published data set that show the increased mTOR1 activity in Treg cells. So therefore, the enhanced mTOR1 activity in Treg cells are important to control the Treg-mediated tumor tolerance. And in the absence of this pathway, the anti-tumor immunity can be enhanced. Having shown the role of REG-A, REG-B small G protein, we also study the Rab, the other branch of the small G proteins. Here we found, again, we can only get the profound immune phenotype when we delete both this molecule, the Rab molecule, which has been well studied in T cell, but then we also found indeed there's a quite extensive genetic redundancy in that we have to delete both the Reg and the Rab2 molecule to drive this loss of the Treg phenotype, and also to drive the mice to early lethality, suggesting the Rab1 and Rab2 are redundant regulators of Treg cell function in vivo. And in data not shown here, we've also established the importance of Rab to link amino acid derived signal through the TSC complex, which is the negative regulator of Rab function. So therefore, altogether, we conclude that REG-AB and Rab1/2 mediates amino acid-induced mTOR activation, while the amino acid, especially arginine and leucine, are crucial to drive and license TCR-dependent mTOR activation. In the next 10 or 15 minutes, I'll switch gears and discuss our work on metabolic reprogramming of CD8 T cell immunity in the tumor microenvironment. As we know, tumor microenvironment is one of the most complex biological systems. It involves highly interconnected cell-cell communication networks, as well as involvement of nutrients, hypoxia, and metabolite as signaling mediators. Adding to this complexity is the intricate and poorly understood metabolic network, which is difficult to study through conventional approaches. Jun Wei and Liu Yunlong, two talented fellows in this lab, decided to take this challenge. They asked, can we reprogram T-cell metabolism to improve adaptive cell therapy for cancer using functional genomics approaches? And that approach is pooled CRISPR screening. To this end, we developed a FOCS CRISPR library in collaboration with John Tench, which allow us to target all of the known metabolic enzymes, transporters, and regulators for a total of over 3,000 genes, altogether around 20,000 guide RNAs. Of transducing the pooled metabolic library into tumor-specific T cells, we pack them into tumor-bearing mice, and then isolate tumor-infiltrating T cells 7 days later. The idea here is, if we can target a major natural regulator of T cell anti-tumor immunity, then those T cells should be enriched in the tumors. Michael Wang and others help us with the bioinformatic analysis, although in this case, the results are so clear. Because all top hit guide RNA are targeting one gene and one gene only. That's known as Regnase-1, which is RNA binding protein with RNase activity. In our validation experiment, T cell deficient in Regnase-1 can be enriched in the tumors at later time by hundreds or even 1,000-fold, and they show much better persistence in vivo. Associated with this better persistence and function, Regnase-1 T cells showed markedly improved therapeutic efficacy in the adaptive cell therapy for E16 over melanoma. This is the original model for our CRISPR screening. And also more importantly, it shows highly improved efficacy toward the BLL that express CD19 as a surrogate antigen in a CAR T cell clinical model. This is in collaboration with Dr. Carrie Geiger and Wenting Zheng. So therefore, loss of Regnase-1 can markedly improve efficacy of adaptive T cell therapy. At the mechanistic level, T cell differentiation state is a known key determinant of anti-tumor immunity. And the conventional thought is T cells with self-renewal and stemless features have better persistence and therapeutic efficacy. But this is at the cost of reduced effector or cell killing function. In particular, recent study revealed TCR-1 positive progenitor-like T cells as critical for sustaining anti-tumor responses. To understand how the better persistence of Regnase-1 knockout cells is regulated, we did a single-cell RNA sequencing analysis in collaboration with Jiang and Tracy. In this case, we found that in the absence of Regnase-1, which is shown in this green, orange color on the left side, those cells showed much higher TCF1 expression. The gene including TCF1 is TCF7. And this can be validated by flow cytometry analysis. They can show the Regnase-1 knockout cell has much higher TCF1 expression. So therefore, Regnase-1 deletion can increase TCF1 expression and the proportion of this TCF1 positive progenitor-like T cells. Interestingly, despite this increased persistence, Regnase-1 knockout cell retain robust effector function. Actually, at the later stages of anti-tumor responses, they can produce even higher level of cytokines and the granzyme B, the cytotoxic molecules, in comparison with wild-type control. So therefore, although the current model highlights that the T cell in vivo persistence and effector function are two reciprocal events, Our data suggests that Recognize-1 knockout can reprogram tumor-specific T cells to gain both features, both the enhanced in vivo persistent and robust effector function. So therefore, we call them long-lived effector cells. While we're quite excited about this finding, a key question still remains. How does Recognize-1 function at the molecular level to control tumor-specific T cell responses? To address this question, Jude and Liu did a truly chaotic experiment by applying second-round CRISPR screening for the genetic rescue of Recognize-1 knockout phenotype. So basically, they co-transduced tumor-specific T cell with Recognize-1 targeting guide RNA plus the genome-wide CRISPR library. And the goal here is to find genes whose deletion can block the Recognize-1 knockout phenotype. This approach is feasible mainly because we were able to take advantage of the massive accumulation of Recognize-1 knockout cell in the tumor. Because in the wild-type context, we probably need many mice to do such experiment. Still, we run into a problem. We simply have too many hits. And in retrospect, this probably made sense because deletion of many of the housekeeping genes would block Recognize-1 knockout phenotype. To narrow down to the specific target relative to Recognize-1, we apply the second criteria to focus only on those genes that are upregulated in the Recognize-1 knockout RNA sequencing data. This is in consideration of the function of Recognize-1 to negatively control gene expression by degrading their mRNA. So applying both layers of criteria, we obtain only 2 candidate genes. And with the transcription pioneer factor BATF immediately causing our attention. We validated the upregulation of BATF in Recognize-1 knockout cells in the data not shown here, and more importantly, co-deletion of BATF blocked the effect of Recognize-1 knockout cell to improve anti-tumor therapy. So therefore, highlighting BATF as a novel Recognize-1 functional target. We further explored the molecular mechanism by which BATF function. Since BATF is known pioneer factor, we did ATAC analysis for chromatin accessibility assessment, and we found that many of the mitochondrial genes showed upregulation in the Recognize-1 knockout cells in terms of the chromatin accessibility, with the approximately 2/3 of them reversed in the Recognize-1 BATF double knockout cells. Consistent with this observation, mitochondrial mass and mitochondrial membrane potential, which are generally associated with T cell metabolic fitness, are upregulated in Recognize-1 knockout. And this upregulation occurs in a BATF-dependent manner. Moreover, ectopic expression of BATF alone is sufficient to drive the enhanced mitochondrial mass and the mitochondrial membrane potential. And we can further validate many of these flow-based mitochondrial features with sequence analysis of the mitochondria-dependent oxygen consumption. So we therefore conclude that Recognize-1 BATF access shapes mitochondrial fitness in anti-tumor T cells. Moreover, our secondary genome-wide CRISPR screening also provide a rich opportunity to identify genes whose co-deletion can further potentiate the effect of Recognize-1 knockout in terms of the enhanced T cell accumulation in tumors. This is important because combination therapy is key to clinical success of immunotherapy. And the top hit here are PTPN2 and SOCS1, and co-deletion of either this molecule in the Recognize-1 knockout background can further enhance anti-tumor effect, therefore providing new opportunities for combination therapy. So therefore, we conclude that targeting Recognize-1 can reprogram long-lived effector cells through the two distinct mechanisms. One is through BATF-dependent mitochondrial metabolism, and the other one is reprogramming the T7 positive progenitor-like cell differentiation. And also from the primary and the secondary screening, we're able to identify novel targets to boost antitumor immunity and also the opportunities for combination therapy. And in this Nature News and Reviews by Ananda Goodrath, this cartoon nice highlight the reprogramming of T cell fate by Regnis1. In wild type cells, the T cells have limited cytotoxic effect towards tumor, but in the absence of Regnis1, the memory-like T cells are reprogrammed, and also this is associated with increased cytotoxic T cells. So ultimately, this leads to the better killing of tumor cells. Moving forward, we are interested in further developing these approaches by building upon the target we identified and using the adaptive cell therapy, especially CAR T cell therapy, to generate innovative strategy towards targeting cancers. So this concludes the scientific discussion of today, including our work in system immunology, nutrient sensing and signaling in Treg cells, and then the metabolic reprogramming of CD8 T cell immunity. With that, I'd like to acknowledge the contribution of my laboratory, especially Hongbo Chi and Nicole Chapman. The first discussion, the project, basically the RAC and the RAP small C protein and amino acid sensing in Treg cells. And Junwei and Ling Yun did the CRISPR screening for anti-tumor immunity. I'm also grateful for the collaboration with Yuming, Jian, and other faculty and staff at Jude. And also I appreciate the collaboration with the scientists outside, including John Dench, Kunliang Guan, and Jason Loxley. And also finally want to acknowledge the funding sources from NIH, including NIAID and NCI. You can see we have a large group of people. Everybody is so nice, although sometimes we can also be very funny. Thank you for your time. I'll be happy to take any questions.
Hi. I hope that the audio is working fine. We have like a small technical problem. Thanks for the presentation. It was amazing. We're gonna go now to review some of the questions that we received from the audience. We remember everybody that if you want to submit questions, we are live, so you can submit in our chat, and we will try to address as much questions as possible. So the first question that I have here is like, how redox situation in microenvironment is important in Treg-mediated regulation?
Sorry, can you repeat the question?
Yeah, the question is how redox situation in the microenvironment is important in Treg-mediated regulation.
Oh, you're referring to the redox
yes.
regulation? Right. Yes, it is very important, although we really don't know, have so far, great deal of information. We do know, for example, it's very nice work from Doug Brenner when they knock out GCLC, which is a molecule involving glutathione metabolism and redox regulation. The Treg lost their function. So this is really going to be an open area for further exploration. Thank you.
Okay, good. And the next question that we have is related to how is mTOR activity differentially regulated in T cells versus other cells?
Right. So since mTOR is a central node to integrate multiple upstream signals, so it can be activated by many of the evolutionarily conserved pathways between T cells and other cell types. On the other hand, T cells have many unique features in terms of their activation and differentiation. So they also have many contact-dependent and unique regulators. One example is our work and others shown that LKB1 is critical for it's a kinase for Treg function, but although in many other cells it goes through inhibition of mTOR, but in Treg cells it's independent of mTOR activator. So therefore, there's both shared and unique pathway involved. And for T cell, we still have a lot to learn. All right, thank you.
Okay, good. And I have like a personal question related to that. It's about the role of glucose contributing to mTOR activation in this Treg. So you observe like the effect of amino acid depletion on mTOR activity in the Tregs. So do you think that glucose is also can have also like an effect in the mTOR activity in Tregs? Like could be like any combination of glucose and amino acid starvation could be affecting even more, or like suppressing, having more suppressor activity for Tregs?
Sure, that's great. So yes, glucose does activate mTOR in Treg cells and also other T cells. What's in between, right? What mediates glucose-induced mTOR activation is unknown. So that still will be an interesting area for further study. So right, again, that's why also we don't know much if we compare. As a consequence, we also don't know much if we compare glucose with amino acids in terms of the intermediate molecule mediating mTOR activation.
Mm-hmm. Right. I have another related question here. So is the effect on amino acid starvation on mTOR regulation in CD4 or CD8 T cells similar to what is observed in Tregs?
I think it's yes and no, in a way, because if we compare, let's say, naive CD4 or CD8, they are, in a way, in less activated state compared with resting Treg cells. That's why the Treg cells have a higher baseline mTOR activation. However, after anti-stimulation, all these different cell types will upregulate mTOR. So that's consistent with the idea mTOR is a signal integrator and for this different type of T cells. Thank you.
Okay. So we have here one more question, and let me check the ones that were already one. Yeah. So how is AG1 activity regulated in CD8 T cells?
Actually similar, almost like a similar, not similar, like analogous to M2 activation. Regnase-1 is such a powerful molecule. It's a powerful molecule to program CD T cell function. So therefore, there's multiple pathways can activate or inhibit Regnase-1 activity. In CD8, Regnase-1 or T cell in general, Regnase-1 can have the regulation at the mRNA expression level, or at the can be also phosphorylated. For example, shown by others by IKK. So we found in the tumor infiltrating CD cells, actually, is the Regnase-1 activity is different from the peripheral or the overall, the T cell in the peripheral. And in that case, because we show that the tumor antigen plays an important role in modulating Regnase-1 activity and also Regnase-1 dependent gene target. So that's quite a fascinating molecule to study further the upstream regulators. Thank you.
Okay, and I have a question here, perhaps like that, I don't understand the total question, but perhaps you can understand better. It says, is T cell effective more than B cells? I guess that is asking like if there is any effect of this mTOR regulation in B cells.
Yes, I mean mTOR is also it's very important for B cells. There's a nice study from multiple groups, including, for example, Mark Boothby, that highlight the critical role of mTOR activated, mTOR function, mTOR activation in B cell responses and B cell function. So it's equally important, in T cells and B cells. Thank you.
Okay, good. So we are reaching the hour of our presentation. I will ask the last question, and then like for people that submit questions and we didn't have time to address, we will send a question to Dr. Hongbo Chi, and he will probably be able in the next weeks to answer directly. But the last question is I lost the question. So, did you find any effect of expression IRG1 with Regnase-1?
No, we don't know that yet. That's an interesting question, but we haven't found that link.
Okay, great. Well, thank you very much to all our audience. Thank you very much for your presentation. And you can join us with our next speakers during today. Thank you very much.
All right. Yeah. Thank you so much, everyone.
Great talk.
All right. Thank you. Thanks for tuning in. Bye-bye.
Bye.